Hey, Miss Chuan, want to meet some geniuses?
Adenine phosphate deoxyribose. This is how the structure of DNA goes.
Anti-parallel with base pairs form a double helix with H bonds they share.
Adenine phosphate deoxyribose. This is how the structure of DNA goes.
Anti-parallel with base pairs form a double helix with H bonds they share.
DNA replicates in many different ways. Without it we die, it's important to life. Isn't this really awesome?
Listen to our easy explanation about semi-conservative replication. First, helicase unwinds the double helix, being the two strands kept apart by single-stranded binding proteins.
DNA gyres reduce tension when unwinding. SSBs prevent hydrogen bonds from binding. Replication focuses where the helicase inserts. It makes two complementary strands that fit.
There's a leading strand and the lagging strand. DNA polymerase builds complementary strands.
It uses RNA primers as starting points and moves in 5-3 direction to make it join.
The leading strand is built super easy. The lagging strand is backwards, so it's super crazy. So it's built in sections called Okazaki fragments, and DNA ligase does the attachments. That's not all. We've got protein synthesis, single-stranded DNA. This is where it begins.
Ribonucleic acid, uracil, naphthyamine, 3 types put together to make a protein. Our RNA makes a ribosome, 2 subunits. tRNA is amino acids, has few bases.
mRNA is a template transporting instructions into the cytoplasm, time for transcription.
Initiation, elongation, termination, cap and tail. Spliceosomes and exons, all introns are gone. This is how we transcript. Initiation, elongation, termination, cap and tail. Spliceosomes and exons, all introns are gone. This is how we transcript. 4 steps, get it right. RNA polymerase finds promoter site. Looking for the C's and A's, unzip double helix, now we gotta elongate.
You're looking for a strand to make mRNA. You're looking for a strand with U as a base.
Synthesize all those genes and stop at the terminate the sequence.
mRNA saved by the template. Time we modify where cap and tail.
5 cap on the start of the primary transcript, poly A polymerase, no degradation. Spliceosomes, no more introns, we gotta put together all those exons.
mRNA, you have a new friend. It's a ribosome, must be translation.
Ribosomes, wherever you are, I'm mRNA, and it's you that I want.
mRNA, you already know I've got the A and the side. What you waiting for?
tRNA, do we have to plead to start remove your methionine?
tRNA, it's time we make the polypeptide chain. You ready? We got so many genes that are regulated. They're always being transcribed and translated.
Housekeeping genes are made all the time, but factors decide when the time is right.
There's 4 types of controls that make this happen. We're going to tell you about them all while rapping.
Transcriptional is the first. It controls the rate at which transcription occurs.
Next, post-transcriptional happens before. It removes introns, splices exons, and more.
Translational controls the rate. It affects the speed of mRNA.
Shh, baby, there's one more left. Post-translational is the last but the best.
Affects the rate that proteins turn active by adding groups to make them less attractive. Now you know controls that regulate genes. Don't you know now how to manage proteins?
Now it's time to move on to something new. Mutations is the next topic we'll teach you.
There's lots of kinds that cause disease, but can all be classified, so listen up please.
Biology class, let's get to the point, 'cause we're on a roll. You ready?
3 different kinds of mutations.
3 different effects on the human nation.
3 different kinds of
3
mutations.
different effects on the human nation.
Silent. Doesn't do no harm. It usually only happens with the introns.
Listen, it changes the base, which changes the amino acid without a trace.
Nonsense. It's by far the worst. It puts a stop codon instead of amino acid first.
Substitution and deletion of a base are reasons for omissions and nonsense mistakes.
Insertion and frame shift mutation can change the acid or its location.
Point mutation causes many syndromes and happens at the base in the genome.
Translocation, use DNA from one side to another in genomes and there.
Induced or spontaneous. Mutations are bad, induced or caused by agents that make you feel sad.
Biology class, all that you've learned will change the lives of all you boys and girls.
All these mutations have to be cured. In the next topic, you will learn more.
Will the real Slim Shady please stand up? I repeat, will the real Slim Shady please stand up? We're gonna have a problem.
I'm the real Slim Shady. Or should I say?
Sim Shady.
Cut it, 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 cut it.
That's one too many strands.
You need to cut it.
Restriction endonuclease.
Cut it, 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 cut it.
If it makes sticky ends,
You need to cut it.
but ends is not the same.
You need to cut it.
Full base pairs this time again.
Cut it. Restriction endonuclease. Cut it.
Recognition DNA. Foreign, not just you wait. Add a methyl group to a base. It's a cynomethylase.
DNA ligase like glue. Uses condensation too. Reproduce sugar backbone. You ain't sick of this track, so
Let's do applications. No enzymes, no hesitations. Gel electrophoresis, I thought you never asked for it.
DNA samples and gels. Currents pull them from wells. The smaller they are, the quicker they move. Minus a plus, ain't nothing new.
But wait up, hold up a minute.
Bacteria has DNA in it.
It's a circle piece, causal plasmid, vectors, host cells. It's a package. Endosymbiotic relationships with their hosts. Their cloning sites have endonucleases. So,
Cut it, 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 cut it.
Genetic engineering, getting bigger every year. Alters DNA sequences, solves problems you fear. Somatropin, somatotropin, the same things? Tree growth deficiency by muscle building. As we sing, we're leaving for now.
say bye to the crew. But we've got one more. Say hello to part 2. TC reaction, y'all don't get it, do ya? 3 steps I ain't playing, it's easy, let me show ya.
Step 1. Denature at 96 degrees Celsius, breaking hydrogen bonds. Separates the strands, working hard, building complementary strands.
Step 2, are you reaching for that tissue? Lower your temperature, attach RNA primers.
How you feel, big and heel. 55 degrees to 65 still.
Step 3, and I'm feeling a shiver. Raise the heat, tack peas, got the lead, okay.
Then extending, we'll holler whenever that's stopped. Making more strands now, we don't need no mascot. Denature, anneal, extend.
Don't forget, that's your best bet. Genetics has just taken a turn. Shout goes out to Human Genome Project, you a real one. Polymorphism detecting differences in day 1. You thought this was over? You thought we were done?
Oh, that's how you feel, man. It's really how you feel, because it's not over yet, and you really gotta chill. Maybe we'll wrap it up, or maybe we want to drill into more biotech. I'm thrilled now.
Let's go!
Now you want to know how far the applications go. PCR, that's a model, you already know.
Forensics, transgenics, therapies, and more. Fingerprints, genetic crops, genetic disorders. Every day, PCR has its way. Peace out, the geniuses are done for the day.
Bloopers! I can look at my spam!
I don't follow your spam.
It's